Morning all, respected Dr. M.N. Patel, Provost, Parul University, Dr. Vandana Patel, Professor and Dean, Doctoral Studies and Research, Parul University, Dr. Abhay Dharamsi, Dean Faculty of Pharmacy and Principal, Parul Institute of Pharmacy, Dr. Guno Chakrabarti, Principal, Parul Institute of Pharmacy and Research, Dr. Lalit Lata Jha, Principal, School of Pharmacy, Dr. Devang B. Sheth, Associate Professor, Class 1, Department of Pharmacology, LM College of Pharmacy, Ahmedabad. On behalf of Faculty of Pharmacy, I, Dr. Anand B. Pithadia, welcome you all in a webinar on challenges in development of vaccines and their safe use in human. I wholeheartedly welcome Dr. Devang B. Sheth, sir, as an expert. Besides his busy schedule, Dr. Devang has given us time to interact with his students and deliver his talk. Let me introduce Dr. Devang, sir. It is my honor to introduce Dr. Devang B. Sheth. Dr. Devang is working as an associate professor, class 1 in the Department of Pharmacology, LM College of Pharmacy, Ahmedabad. He has completed his M.Pharm course from Gujarat University and Bachelor of Pharmacy from Bir Narmad, South Gujarat University. He has completed Doctor of Philosophy from Saurashtra University. He has 15 years of teaching experience, including 7 years of postgraduate teaching. He has published around 13 international publications in various reputed journals. The achievements include here he has obtained first prize for poster presentation in Silver Coin Annual Conference in IPS Gujarat Chapter Ahmedabad 2006. Received gold medal and from Professor M.N. Jindal Prize 2006 Ahmedabad branch for IPS. He was co-convener in 27th Annual Conference in Indian Pharmacological Society Gujarat Chapter held at RK College of Pharmacy, Rajkot 2008. He was chairman and abstract committee in International Conference of IPS, Indian Pharmacological Society 2015. He has coordinated very different events related to the field of pharmacy and pharmacology and served as peer reviewer in many international journals. So with this, I would like to request you, sir, to start this session.
Thank you very much for nice introduction, Anand sir. I thank the organizers, the administrative, because of that the members of Parul University, Dr. Abhay Dharamsi sir, Director Madam Sri Dr. Lalit Lata Jha Madam, and Dr. Anand to provide me this platform to share my knowledge. Without wasting my time, let me just start with the topic of today's discussion. Sir, kindly confirm that is my slide visible to you?
Yes, visible and you are audible, sir.
Thank you, sir. Thank you, sir. So, upon completion of this webinar, you will be able to define vaccine, describe various types of vaccine. You will be able to understand basic knowledge of immunology relevant with vaccines and discuss the generation of immune response against the vaccine. You will be able to state the importance of vaccination and derive measures responsible for effectiveness of vaccination. Apart from that, you will be able to narrate key stages which are required for the development of a vaccine, and you will be able to compare the similarities and difference in the conduction of clinical trials for a drug and a vaccine. By learning through, you will be able to identify the challenges observed in the development of vaccine, and then finally we will briefly go through the basics of the adverse events following immunization. So these are the learning objectives of today's webinar. Although the timeline is 1 hour, I will try to discuss each of this in brief. So we know that every day we are exposed to a large number of infectious agents. However, only a few of these exposure results in a disease. And we know that. Why? It is because our immune system is working properly. Right? So to immune is to have freedom, right? To have an exemption from the disease, right? So it is the system which protects the body from the disease. You know that the body has a proper recognition system. So you have specific MHC molecule, major histocompatibility complex, right? Which are present on the cell surfaces. And if other than these molecules are present in the body, then they are considered as an antigen. So those substances normally are proteins, but many a times it could be polysaccharides also. And these substances, once entered, they are considered as an antigenic. They will stimulate the production of antibodies. Antibodies, we all know that is a complex glycoprotein which is secreted by the plasma cells, a special type of a B cells, right, in response to an antigen. Wherever there is an antigen antibody reaction, we call it as an agglutination. Apart from this, these are some of the terminologies which may be useful in the webinar, which is we are discussing right now. So passive technique of immunization, you have antiserum, that is you have injection of antigens in animals, and that animal may produce a specific type of antibodies which are isolated from the serum of that animal, and that is referred as an antiserum. And anti-rabies serum is one of the example. Similarly, some cases of bacterial infection, you have exotoxins which are predominantly producing the pyrogenic responses or the complications of a disease. Such a case, we do modify this exotoxin by some special procedures, say for example passing the formalin gas, and thus one which is similar to toxin, that is what we call as a toxoid, right? So that toxoid is safely used in immunization against the disease. Such type of preparations are again available for because the treatment of for prophylaxis of tetanus and diphtheria, right? So these are called as a toxoid. So now, what is vaccine? So vaccine is a substance which is prepared from the causative agent of a disease, that is the bacteria, virus, fungus itself, or maybe it is a product of that microorganism. Say for example, sometimes we isolate the proteins, right? You call as a subunit type of an vaccine, right? So it's a part of because a bacteria that is being used, or maybe it could be a completely synthetic substitute. Like say for example, we have prepared in the laboratory and then we are introducing as a vaccine. So it could be either causative agent, product, or synthetic substitute. Primarily, it is used to produce the stimulate the production of antibodies, and by producing the antibodies, we provide the immunity against one of the several diseases, right? Sometimes against one disease, sometimes if it is a recombinant, maybe it could be the single vaccine is effective for the protection against many diseases. And then it is very important that when you are introducing this antigen itself, it is not producing the disease itself, it is not inducing the disease. This is what a legal definition of vaccine is. So in short, we are intended to provide the immunity against the disease. Now, the importance of vaccination can be considered or can be understood in a way that those countries where the high vaccine program coverage is observed, then the diseases which are prevalent previously, especially in case of childhood, right, where they are responsible for mortality, they have essentially disappeared. These are some of the data. You can just see that the disease like measles, diphtheria, polio, this all definitely, once the vaccination concept was introduced, then there is a drastic decrease in the prevalence of this disease. This shows the importance of vaccination, right? Even there is one very important observation in case of pertussis disease, bacterial disease caused by Bordetella pertussis. Vaccination was introduced somewhere in 1950s, which resulted in the reduction in the prevalence of the disease. But then there was a time, 1970s, then vaccination was reduced for this the disease, and then you can just see that there was a rise in the there was increase in the prevalence of the disease. Further, once again implemented strategically by various the governments, then again you can just see that now the cases of pertussis are very low, right? So as per WHO, we say that because of the current immunization program, is approximately 2 to 3 million lives are being saved. These are the data of WHO, recent data of WHO, right? So previously, if you just see that mortality is associated with the childhood phase was very high, the morbidity is associated with some of the disease was very high. But now, yes, with the effective vaccination program, yes, we have definitely a reduction in that morbidity and mortality, right? So you can just see that comparison, right? So marked reduction in the mortality of the children less than 5 years of the age globally, from 93 deaths per thousand somewhere in the 1990s to 39 deaths in the per thousand live births in 2018. So the question should arise in your mind, that is there a need to understand immunology for developing vaccines? Because if you take few examples, histological examples, say for example of smallpox, right, or maybe anthrax, right, these were very initial vaccines, right? So Edward Jenner, who first introduced the vaccine, it was an empirical research, it was based on observation, right? It was not a proper immunology for on the basis of which vaccines were developed. Similarly for chickenpox, maybe for some disease like anthrax, Same type of strategies or that empirical research, right, lead to the discovery of vaccine. But yes, definitely, if you just see the present scenario, you have to consider that understanding of immunological basis of vaccination is must, without which definitely it will be very difficult, because the hurdles which we are having right now are are much tough as compared to the previous one, right? We have hard to target pathogens like tuberculosis, mycobacterium tuberculosis. We have some pathogens which are continuously variable, antigenically variable pathogens. HIV, you can take example of SARS-CoV-2 also, current it is a pandemic condition. That also is highly variable antigenic pathogen. Then you have some outbreaks which have threatened the global health security. Hello. Do I need to change anything?
No, sir. Please continue.
Okay, fine. So, to control the outbreaks that threaten the global health security, say for example, Ebola, COVID-19, these pandemic situations have definitely threatened the global health. Right? You can just see the because there are two different properties which normally we require, right, for to understand a disease, right? How deadly the disease is and how fast it spreads, right? RO number we consider. So in both cases, definitely, right, this is the WHO data, right? So death rate is somewhere around 1.34% right now. It is variable across the globe, right? In different countries, because the values are different, right? But then definitely fatality rate is high. Similarly, the spread also is comparatively high, right? It is definitely not as high as what we observed in case of Ebola or Middle East respiratory syndrome, but still the way, right, because it is, we are now knowing that it's an aerosol transmission, right, that is possible for the COVID-19. So such a case, such type of a disease, definitely if you are knowing what is the immunological aspects, then it is definitely it will be easier to develop, and that is what we observed in the last year, right, that we have very fast a development of a vaccine for COVID-19, right? It was that record break, because the time you can just see that satisfactory vaccines are available in the global market. Also, if immunology is concepts are clear to you, you will be able to generate or revive the immune responses in the aging immune system, right? Especially for the growing population in the older adults, right? On the geriatric population, you know that the mortality rate in case of COVID-19 is very high in geriatric. So such a case, definitely you can understand these all if your immunology concepts are clear, right? This is the current situation of SARS-CoV-2 infection. Daily cases, which was previously in first wave, were comparatively low. A sharp rise in the cases as well as the death have been increased. These are the government data that a death rate of 1.34% is been observed till date, right? This is last evening data that I have collected. So still it is a very high death rate you can consider for the disease. Now a question should again be there in your mind, that what type of immunity does vaccination provide? Right, so I've given different parameters. Definitely we are taking it from outside, so it is acquired. You are taking it and then the body is generating the antibodies. So when you have generation of antibodies from your body, right, it's not a preformed antibody you are taking, so that's why active. And because you're taking it artificially, right, when you have a natural infection, it is a natural type of an immunity you can consider. But when you're giving it through the injection, it is artificial type of. So it is acquired active artificial immunity that you can definitely consider for the vaccines which we are taking, right? So just going through very basics of immunity, you have two types of immunity, acquired and natural. Innate immunity or natural immunity, you have lots of barriers like physical, physiological, cellular, cytokines, right? So these all barriers definitely do play a role, and they have it's a first line of defense that you can consider, right? But then you have very specific type of an immune system where you can have active or passive type of an immunity being given to a person. Active immunity, definitely you can have a natural, maybe after a clinical or subclinical infection, you know that person is having a infection, then definitely sometime it is a lifelong memory, it's being created, or maybe say for example at least for some tenure the memories are created and thus gets protected from the further exposure of that pathogen. Similarly, vaccines, we do the same, but then it is by the artificial. Passive, we directly administer the antibodies. Natural ways of maybe say for example the protection to the child by the mother through the transplacental passage, or maybe the initial milk, which is referred as the colostrum, right? This can provide the immunity to the child. Or maybe you can say that you have artificial type of an a passive immunity, which you can give by use of some serum, right? Or maybe say for example some globules, right? That is again an anti-serum, right? So homologous means it is from the human only. right? Say for example, in some cases the antibody titers are comparatively low. So what you have to do is you have to go for a pooling of antibodies, right? You have to concentrate the serum, and that pooling of, because that homologous antibodies can be given. Such type of strategy is effectively used in treatment of hepatitis A and B cells. Similarly, hyperimmunoglobulins, if the amount of antibody titer is higher, then you have a hyper immune type of globulins. This is again used in post-exposure prophylaxis of hepatitis B, rabies. We know that once a rabies or maybe a dog bite or a cat bite is the case, then you can use such type of a passive immunization technique. Heterologous hyperimmune serum, you have same thing, but then you have the immunoglobulins which are derived from the animals, not from the human. That's why we call it as a heterologous hyperimmune type of a serum, right? So these are passive immunization techniques which are also being followed somewhere for the prophylaxis as well as treatment of some of the diseases. Now let us understood how does vaccination work, right? So what does vaccine do is vaccine exploit the extraordinary ability of your highly specific, highly evolved human immune system, right? So that is responding and remembering the encounters with the pathogens, right, or the antigens which they have received. So you know that there are two types of lymphocytes, B and T cells, right? These cells you have plasma cells and memory cells. Similarly, in case of T cells, you have helper T cells, you have T suppressor cells, memory cells, and cytotoxic T cells. So these T cells, that is cytotoxic T cells, provide cellular immunity, and then you have bone marrow cells which maybe can differentiate into plasma cells and then lead to the generation of, because of that, the immunoglobulins, that is IgG, A, M, D, E. Because the immunity is provided in the humor, that is the body fluid, that's why you call it as a humoral immunity or antibody mediated immune system. So 20 to 30% of your third line defense, right, very specific defense, is such type of a defense, while because that 60 to 70% or major is the cellular immunity. When it comes to vaccine, normally we measure the immunity in terms of antibody mediated response. We measure the seroprevalence in the individuals, right? But then at tissue level, the cellular immunity also is very important, right? Only thing is we are not able to understand it properly, or we are not able to directly measure the immunity, because T cells activity you cannot directly assess. You have access to the blood samples, you don't have the access to the tissue fluids, right? So that is the reason why we more focus on the antibody generation. But yes, tissue mechanisms that is related to cellular immunity are equally important, right? This is the cell that is helper T cells, which is characterized by the presence of CD4, cluster of differentiation 4. So when CD4 is present on the cell surface, we call it as a CD4 plus cells, that is your TH cells, T helper cell. This T helper cells not only helps in the clonal proliferation of the B cells, but also helps in the clonal proliferation and activation of the cytotoxic T cell. Thus is a very important mediator in the generation of an immunity, right? So these are the cells of bone marrow. Further classification, definitely I have not shown here. You can further classify these T cells on the basis of the expression of cytokines, right? Say for example, Th1 cells, Th2 cells, maybe in case of lung, Th17 cells. These are very important cells, but maybe you can say that if you are doing some type of research, you can go in the details of this thing. So this is the one. You have special types of cells in your body, which you call as an antigen presenting cells. Once been activated by the T helper cells by the release of cytokines, they do recognize the antigen, and then they undergo the clonal proliferation, right? Under differentiation, you can have plasma cells, which I told you, that is responsible for the production of antibodies, and you have memory cells, right? Some of which are short living, some of which are long living, and they will be stored in bone marrow. Whenever second encounter is there, they will be able to memorize, and they will be able to produce the antibodies within a short time, and thus effectively able to counterattack the pathogen antigen which is present in the body, right? So characteristics of acquired immunity: it is pathogen specific, it is characterized by the memory, which is very important characteristic. So primary response is always a low intensity response, right? Because we have encountered that pathogen for first time. So when, say for example, you have an actual disease, this is what happens. Your body does not have memory, so it will not be able to immediately initiate the antibody response. It will not be able to initiate the cellular immunity response, because it is the first time we are encountering, right? So vaccine does the same, but then vaccine, you have antigen, but you do not have the pathogenicity, right? That antigen is just giving an alarm to you, or is just notifying you. So such type of an molecule can come and can produce some type of an problems, abnormalities, So that is what vaccine is doing. It is generating the primary response without actually producing the disease, right? That is what you call as a vaccination, right? Secondary response definitely is an recollection of previous existence. So you have memory cells, and that memory cells will be effectively able to generate the antibodies, which will be able to produce a highly intensified response. Why? Because your body have memory of the first encounter, right? So this is what happens. You can just see that first antigen response, primary response is slow, it is low intensity, but yes, wherever second exposure is there, you have a second response which is more intense, more rapid, and thus effectively is able to deal with the, if not able to prevent, at least the symptoms. You can just take that you can have minimum symptoms, minimum abnormalities, minimum pathological complications, right? So this is called as an anamnestic response. This is what we are intending to produce through the vaccination, right? So this is how, because of that molecular mechanisms, or because of how the generation of immune response to a vaccine can be produced, can be learned. You can just see that vaccine once introduced, normally it is the muscles which are exposed. Sometimes it is an oral route also we can consider, but let us consider that if it's a parenteral preparation, then in such cases vaccine antigen will enter. You have lots of cells which have a specific type of an molecule which is called as an MHC2. MHC2 is major histocompatibility class 2. You have normally three types of MHC present in the cells. MHC1, right, that is present in all nucleated cells, so virtually all cells except RBC and platelets, right? Apart from that, all cells have MHC class 1 molecule. But MHC class 2 is present on only specific cells. The cells which have this type of an molecule, they are called as an antigen presenting cells. So say for example, in case of tissues, you have dendritic cells. Dendritic cells so called because they have dendrites, they have lots of processes, right? That's why you call it as a dendritic cell. So these dendritic cells, they are able to recognize this vaccine antigen. If it is a live attenuated vaccine, then they will be able to directly recognize the antigen, and the immune response is very good. But if it is a dead vaccine, if it is a part of the bacteria or virus, that is what you call as a subunit vaccine, such a case the immune response is comparatively low. If it is low, you need to create that danger signals with the use of adjuvants. Right? Say for example, alum is one of the common adjuvants. You have lots of advancement in the adjuvants also, but then adjuvants are commonly used in the vaccine formulations, right? So that is to increase the immunogenicity response, to increase the recognition, and they do activate this PRR. PRR is, you can say, that pattern recognition receptors which get activated by these adjuvants, and then they will activate these dendritic cells. Once activated, after some time they will be, you can say, traffic to the draining lymph node. Once they're in the draining lymph node, you have the recognition. You can just see that MHC class 2 can be recognized by T cell receptors. You have T cell receptors, right? And then you have B cell receptors, right? B cell receptors will be able to recognize the soluble antigen vaccines, right? So they will recognize and they will start producing the immune response, right? MHC class 2 I already explained to you, but then it is very important to understand, right? It's actually a gene, that is MHC HLA complex, which forms the MHC antigen, and each of us have a characteristic antigen, and that's why in case of organ transplantation there is a rejection, because MHC for a particular person is a specific one, right? If it is a twins, identical twins, right, monozygotic twins, then and then the 100% compatibility is there, otherwise not, right? So MHC2 is different, and that's why immune response, again, that antigen is bound to happen, right? It is very clear to you. So this definitely, once T cell receptor recognizes antigens which are expressed on the cell surface, right? Your dendritic cell processes, and a part of antigen is expressed on the cell surface, and that is creating the release of cytokines. Once cytokine release is there from the CD4 T plus cell, that is T helper cell, it will activate CD8 plus cell. CD8 plus cells are cytotoxic T cells, where effector T cells or memory T cells be generated. Similar way, B cells, you have plasma cells which will produce the antibody, and memory cells. Some of these memory cells will be stored in the bone marrow, which may have a long life, and then again, you can say wherever second encounter is there, they will be able to produce the antibodies in a very high manner. This is a general pathway that how immune response is generated against a vaccine, right? So definitely antibody levels whenever in the circulation after the primary vaccination, they are below, you can say, a level which is required actually for the protection. In such a case, immune memory is a very important feature of vaccine-induced protection. Whether this immune memory can protect against the future pathogen encounter, it will depend on some of the properties. Let us check that. First, what is the incubation time of infection? It is possible that the generation of second response antibodies, it may take some time. and incubation period of infection, that is seroconversion, is short. Such a case, effective protection is not easily observed, right? So definitely depends on the incubation period. Second is what is the quality of memory response. Sometimes, say for example, in case of dengue, memory response is generated and is executed only for 1 year. So after 1 year, again, if you want to be protected against dengue, you need to again take a vaccination shot. In USA, influenza shots are very common, right? They do not go for such type of an vaccine shot for an effective protection against the seasonal virus, right? And also you need to check level of antibodies which are induced by the memory B cells, right? So there are some vaccines which even after a single dose gives an effective protection, and then on the contrary, you also have some vaccines which even after 2 doses will not be able to produce effective protection. So how does this happen? Let us check few details. You can just see that I have given this vaccine, for example, yellow fever. Once I have given this type of a vaccine, such type primary immune response is of high intensity, and then you can just check that protective antibody level is always maintained above the MEC level. If such type of an immune response is produced with vaccines, then you can consider that it's a lifelong, because that protection that is provided by your vaccination, right? So this is very good example. Yellow fever you can just consider, right? Similarly, you can just see that this is, you can say that another case where exposure of pathogen, you have a very short incubation period. Let's say for example, Haemophilus influenza type B. Such a case, there is an insufficient time to raise the protective antibody level, and thus effective, you can say that protection against the disease is not possible. You need to give a booster dose again, right? Or maybe you can say at periodic interval, so that effective protection can be achieved. Otherwise, it will not be achieved. In some cases, definitely long incubation period is there. Say for example, hepatitis B. In such a cases, definitely yes. After giving the vaccination, even if there is an exposure to the pathogen, effective protection is provided because protective antibody level is achieved after the vaccination, right? So such is the very important, because of the immunological concepts you can understand. This will help you to understand, let's say, why the Johnson vaccine, Janssen, right, is a single shot vaccine effectively been responsible in COVID-19, while in case of Covishield, Covaxin, or maybe the Moderna vaccine, you need to give 2 doses, right, or maybe multiple doses to produce effective protection against the COVID-19. Herd immunity is what we are trying to actually produce with the vaccination, right? So that is what is also government has been trying to do. You can just see that if no vaccination is a case, definitely the prevalence or the spread of disease will be very high. This is, I guess, you can say that current scenario with a few persons who have been diseased, few persons who have taken the vaccine. Still, there is a chance that those who have not been vaccinated, there is a high chance that disease may be spread to the others. While if effective vaccination is a case, you can just see that disease transmission can be stopped, right? And that is what we are trying to achieve right now, right? Maybe after 1 year or 1.5 years, we'll be in a stage, right, where effective a herd product protection is being produced to a large population, right? So this is what a vaccination is actually intended for. So after learning how the immune response is generated against this vaccine, now let us check what type of different vaccines are available in the market, right? So you actually divide in 2 platforms, either living types of vaccines or dead types of vaccine, right? Living, you have only one type, that is live attenuated, or what you call as a weakened or inactive type of an vaccination, right? So what here we are doing is we are making that pathogen, either bacteria or virus, a very weak. So it is just able to generate the immune response, but it is not able to produce the disease. That is what you call as an attenuation. Such type of an attenuation can be produced by repeated culturing. Say for example, I will give you an example of measles. Measles viruses were isolated from a child who was suffering from the measles, and then there was repeated culturing in the tissues for 10 years. And after that, they observed that virus has been weakened, right? Say for example, someone gives you food and then will not ask to do anything, right? And this process continues for 5 years, 7 years, and then suddenly someone says that you have to do a work to gain the food, right? So you will not be able to perform the work. Same thing happens with the viruses, bacteria. That's what called as a repeated culturing technique. But this type of techniques, it requires a prolonged duration, right? You require a prolonged time. Such type of a scenario is not useful if a pandemic is being observed. Say for example, in case of COVID-19, a sudden prevalence has been there, a pandemic situation is there, though you will not be able to use this type of a technique. So what techniques you can use? You can go for a codon pair de-optimization technique. So what you do is, you know that there is a triplet responsible for an amino acid, but there are many triplets which can assign single amino acid. So they do modify these codons, and once they have been modifying these codon, they are optimizing this codon, they will be able to generate some strains which are having the production of proteins, but then they are not able to produce the disease, right? So mutated strains, codon the pair de-optimization techniques are used to produce. That's why I'm saying that immunology concepts are very important for effective development of vaccination, right? So these are some of the vaccinations which have been licensed using this technology. Of course, smallpox was the first one which was been introduced as a vaccine, and it was the first one which was a live attenuated type of a vaccine, right? Similarly, cold kill organism, you can just have a use of heat or chemicals, and you can say that such type of vaccine can also be effectively responsible for generation of immunization response. But yes, wherever you are using killed type of organism, the immune response is normally low as compared to live attenuated vaccine. So such a case, use of adjuvant is compulsory, right? It is very simple concept that more resemblance with the original pathogen, very good will be the response, immune response, better will be the immune response. So say for example, if I am using a killed organism, it is definitely not resembling the pathogen. It is not closely resembling to the pathogen. So such a case, definitely immune response comparatively slow, or you said low intensity response is there. That's why use of adjuvant is definitely advisable in such type of application. Toxoids, you have modified chemicals which are you said exotoxins, and that exotoxins, say for example, in some cases of bacteria like tetanus, they do produce pathological condition with the use of chemicals. Exotoxin is a major route by which they produce the pathology. So such a case, you need to give some preparations, vaccine preparations, which are chemical modification of those toxins, and that is what you call as a toxoids. Diphtheria, tetanus toxoids are widely available in the market. Similarly, you can use part of some of the bacteria, normally a peptide, but sometimes polysaccharide also. Remember, wherever you are using a polysaccharide type of subunit preparations, you need to conjugate it with the protein. That is also called as a conjugate vaccination, right? Conjugate subunit vaccination. So, and this further can be purified with some of the biotechnological processes. Recombinant protein is also available, and that definitely, say for example, in case of COVID-19, Novavax subunit vaccines are available, which are using surface antigens only. Right? So, say for example, hepatitis, meningococcal, pneumococcal, such type of vaccination are available right now in the market. Virus-like particles, they are just having surface proteins, they are having the cell membrane, but they do not have genetic material. Such type of particles are also being used in producing the immune response. Say for example, human papilloma virus right now is been licensed with this type of technology. Some gram-negative bacteria, they have vesicles, right, which are released from their membrane. This type of vesicles, they do provide toxins, or they do have the genetic material which can be responsible for pathological responses, and maybe say for example, they are also important in bacteria to bacterial communication, bacteria to human pathological complication. Such type of an outer membrane vesicles are effectively used also for the delivery of genetic material which are having the antigenic protein. So such a case, meningococcal group B type of a vaccine is been developed and is being available for such type of use of such type of technology. As I told you, in case of polysaccharide, the immune response is very weak. Such a case, carrier proteins are always used to produce the immune response. Even in case of some antigens, you use viral vector vaccine. Again, in COVID-19, such type of preparations are being used. Covishield is one of that. So what you do is you have a non-replicating viral vector in which the antigenic material is being administered, and then effectively is able to produce the antigenic response. So Ebola was the first one to be produced by using such type of a platform, right? So viral vector, even synthetic nucleic acid vaccines are also available. DNA vaccines, RNA vaccines, they do transcript and translated into proteins which may produce the effective. SARS-CoV-2 definitely is the first one which was licensed. Many vaccines for animals, veterinary vaccines are available with the use of DNA and RNA vaccines. But then in case of human, the SARS-CoV-2 was the first one to get approved from the US FDA and EU. Right. Similarly, bacterial vaccine we are working on. We don't have any vaccines used with the use of such type of an platform. Similarly, APC, antigen presenting cells, you don't have such type of vaccines, but yes, it is there in the experimental level, right? So let us just revise, because COVID-19 pandemic is going on. Let us revise these all vaccines with the use of examples of COVID-19 vaccines. DNA vaccine, as we know that some genetic material, synthetic vaccines, right? It has been transcribed to the mRNA. Further will be, so these are very safe type of vaccines which have been claimed because a specific antigen is been produced which will specifically stimulate you. So it is not a whole bacteria which you are, a whole virus you are administering. You are just part of The antigenic determinant protein that is only been expressed, that is being given in the form of a DNA and is being given to the that person. Similarly, in case of RNA vaccine is more safer because it will not never go into the nucleus. Such a case, long-term problems associated with the mutation in the genomic DNA, they are not associated. Only thing is RNA, it is liable to be degraded by various RNases. Such a case, you need to give by some of the novel technologies, like say for example nanoparticle liposomes you can use for effective penetration of RNA vaccine into a person body, right? Similarly, viral vector, you can just see that a non-replicating viral vector where the genes have been introduced, DNA has been introduced, and then effectively can produce an immune response. Subunit, you just specify, you purify the proteins, and those proteins, they have been used to produce the immune response. Live attenuated, you make that virus being very, you maintain the immunogenicity, but then it is not able to produce the disease. So that is your live attenuated type of vaccine. Right? Inactivated vaccine, that is either with the use of heat, or maybe because there's some heavy metals, or maybe because with chemicals, the virus or bacteria is being killed. Right, so in that case, definitely it is still be able to produce the antigenic response. Also, virus-like proteins without DNA, just a surface protein, looks like a virus, but it's not an actually virus, can also be used. And with the use of some detergent, you can have a split of the virus, which again can be effectively used for purpose of the immunization. Even passive immunity has been tried for COVID-19. Monoclonal antibodies, polyclonal antibodies are available. Even plasma therapy, a lot has been discussed in the early phase in COVID-19 treatment, or mRNA-induced antibody is also a passive type of immunity which can be generated effectively, right? Is being used some cases of the COVID-19 cases, right? So it's a simple question. Maybe you have such type of a question. So that what type of vaccine should I go with? Live attenuated vaccine or killed inactivated vaccine? So yes, definitely there are pros and cons which you can consider for both types of vaccines. Live attenuated vaccine, yes, you can produce a very good response with a single type of a dose of vaccine, right? And the duration of immunity, you normally have a long immune response being produced, right? Normally you have IgG type of antibodies which are generated with live type of vaccination, but yes, there is a chance, rarely, that a live attenuated vaccine can revert to its original form, right? Say for example, cases of polio, rarest of the rare, but then some cases of polio vaccine, it is reported that yes, it may revert to its original form, and the disease itself can be produced by the vaccination. While in case of dead vaccines, they are completely safe, right? So it is not possible that it will revert to the original virulent type of pathogen. So it is safe and stable, but yes, you require multiple doses always. Second thing is you need a high dose of this type of vaccines to produce effective immune response, because the immune response is always of low intensity, right? And need of adjuvant is always a case in case of killed inactivated vaccine. Right, so this is overall comparison of live attenuated vaccine and dead type of a vaccine. Right, normally you use such type of a comparison. Say for example, when vaccine slots are available for Covishield and Covaxin, many students or many colleagues of mine, they do ask me that what type of vaccine I should go with. Definitely now you have more options, like Sputnik is available, Moderna vaccine is available, maybe in short time Zycovid also will be available. So definitely you have multiple options, but then whatever type of vaccine is available, if it is available to you, you definitely should go for vaccination. That is what current guidelines, that is what the current need of time is. Definitely you can go for a comparison. This may take a separate lecture, that which one is better, Covishield or Covaxin. Irrespective of all factors, definitely you should go for vaccination. Now let us check the key stages to develop a vaccine. Definitely for developing any molecule, right? Definitely it's a part of biologics we are discussing. So you have basic research, in silico, in vitro research, then come to the preclinical research, and then definitely the clinical research that normally we discuss. So same scenario is the case with the vaccines, but yes, when you consider the vaccines, right? So definitely you need to consider some specific criteria, because we are intending to give to a large population, or almost all people of the country, right, or continent. Such a case, safety is very important, right? Say for example, in case of drugs are going to be taken by the person who are diseased, who are having some problems. Vaccine is going to be taken by a person who is normal, who is a healthy being, right? So such a case, definitely safety is a prime control. Second thing is efficacy. Definitely has to be a case, because that is what it is being intended. That definitely you can have efficacy ranging from 60% to 90%. No vaccine gives 100% protection, mind you, right? We normally have the concept that once it is a vaccinated, we are vaccinated, we are completely safe. But yes, maybe last 3-4 months we have learned that it is not a 100% effectiveness that it can be observed with a vaccine, right? That is not possible with any vaccines which have been existent even before the COVID-19, right? So that definitely you need to consider, right? Also, you need to consider some special procedures like step down process. Right now, definitely COVID-19 is intended to be given to the whole population, but normally vaccines are intended to be given to a pediatric population. So when it comes to the clinical research, you need to consider that you cannot initiate a clinical trial in the pediatric population directly. You need to you need to use a step down approach, right? So let us check the criteria which need to be considered for advanced development. Say for example, toxicity data, you need to consider that in initial studies, in the in vitro studies, in the preclinical studies, whether the toxicity has been produced or not, whether how you will be able to utilize this toxicity data into from animal to the human. What is the nature of immunogenicity response? It is just a B cell memory being produced. It is a T cell memory being produced. Say for example, in case of tuberculosis vaccine, right? Definitely the T cell response is generated in a very good amount, right? So such type of you can say molecular changes you need to learn, right? You need to understand the nature of this response. What will be the possible impact on the public health, right? Because there are some adverse effects, maybe a minor one, maybe a severe one, right, that are going to be produced. So what will be the impact, because you are going to give in a large number of public, right? Similarly, what is the chance of acceptance by community? You need to understand the socio-economical, cultural type of an a characteristic, and then you will be able to increase the acceptance in the community. Cost effectiveness has to be there. If the vaccine is costing somewhere around thousands, people will not accept, definitely, right? So cost effective, and is there a pre-existence of a vaccine with a satisfactory risk benefit profile? Then what is need of developing new vaccine? You need to consider this all thing, and then you need to develop a vaccine, right? So when it comes to drugs and vaccine, there are lots of differences. Normally people consider that they are same, but yes, there is a difference. In case of drugs, normally you take a healthy volunteers, or in case of phase 1 and phase 2 trial, in case of phase 3 you go for patients. While in case of vaccines, normally you take healthy volunteers throughout the clinical trials. Logistic challenges in case of drugs are normally less. In case of vaccine, it is stringent, especially if you are talking about the DNA and RNA. You require a deep freeze, you require -40, -80 type of a storage, which definitely in case of poor or developing countries is not feasible. Step down approach, rarely practiced in case of drug, because it is intended to be given in an adult population. While in case of vaccine, remember, they are normally intended to be given for pediatric purpose. That's why commonly it has been practiced. Normally it is given to an adult population, then to 12 to 18, then you have to go for 5 to 12, and then 1 to 5. Likewise, the step down, this is the age group which we normally go through. Number of volunteers required for drug normally is less. In case of vaccine, definitely huge number of volunteers, very large number of population you need to require. That's why development of vaccine takes a longer time as compared to drugs. But yes, definitely because of the change in the policies, also the efforts of pharmaceutical companies, and a huge effort from the scientists, it is possible that COVID-19 vaccine is available within a short time and with a satisfactory efficacy and safety profile, right? So additional challenges, definitely you can consider. There is a chance that co-immunization is possible. If you are giving 2 vaccines simultaneously, there is a chance that co-immunization may happen. The presence of adjuvants, again you need to consider that what is the safety profile of that adjuvant that you are administering. There is a potential, because a risk that cross-reactive antibodies may generate, immune complexes may be produced, right? Say for example, there are some pentavalent, trivalent type of vaccines. So whether you will be able to give this together or not. Sometimes if previously you have taken a vaccine, after what time second vaccine should be taken, this also you need to consider. Normally it has been recommended that you can take 1 month before or after the vaccines, if the previously studies are not available. And then definitely pre-existing antibodies. Say for example, person is already been infected, or maybe is having the antibodies from the maternal side, then what will be the immunological response that you have to predict? These all additional challenges you need to consider when you develop a vaccine. Also, lag time of onset, persistence of antibody, memory, what is the memory response being produced, what is the seroconversion rates, induction of immune memory, that also you need to consider as a part of kinetics of immune response. Yes? Okay, so
participants are requested to mute their mic.
So that case is definitely components of immune response. May also vary as per the mode of delivery, that is oral or parenteral route, and then definitely you need to consider the quality of antibody. Even if antibody titer is higher, does it having specificity? Does it having avidity? That also you need to consider, right? So it's not just a production of antibody which will assure you that it will be producing the effective effect. So these are different because of that phases of the clinical trial. This definitely you must have learned somewhere. I'm not wasting my time. Phase 1, phase 2, phase 3, same thing is there. Once it has been approved with the phase 3, then it will be available in the market, and then you have still continuation of the studies with the phase 4 clinical trial, post-marketing surveillance. You can definitely. This is the timeline. You can just see that vaccines like polio, you have a whole century or many decades, 7 to 8 decades, been devoted to develop a vaccine, right? And this is true for most of the vaccines which have been developed yet. Yes, but with the efforts of scientists and with the policy level changes which have been recognized, and the multicentric trials which have been conducted within a very short time, and with the use of biotechnological and immunological concepts right now we are having, with the science, the way they have progressed, definitely it is a very satisfactory progress we observed in case of COVID-19. Within span of 1 year, we were able to develop a vaccine. Right? Previously, it was a case of Ebola where within 4 years, we were able to develop, which was considered the fastest vaccine, right, at that time. But then, yes, definitely, coronavirus vaccine has broken all those records, right? So, the development of vaccine is not an easy task. It requires lots of efforts, and this is what we have observed in last 1 or 1.5 years. But yes, definitely, how do we measure the effectiveness of vaccine? Normally, we consider endpoint is protection from the disease. But apart from that, reduction in the severity of the disease, reduction in the rate of hospitalization, reduction in the complications which are related to the disease, all of this you can consider as a measure for effectiveness of vaccines. So this is what we have learned in the the webinar that is been we were organized. Global vaccination coverage definitely in world is still been lacking. We discussed a lot about the availability of COVID-19, but then we have availability of many vaccines. But still, you can just see that vaccination coverage is still not above 90%, right? They say the old disease like polio, TB, still it is around 85, 88%, right? When you see about India, still it's good number, but still you need to achieve almost 100%, right? Then and then a herd immunity could be developed, and then the infection rate, because the prevalence of a disease can be reduced, right? This is what in India the vaccination coverage is there. These are WHO data that I have taken. The same thing is there in US. Definitely in developed countries, the vaccination coverage is comparatively higher, right? So definitely this is all because that we'll go through the specific disease with regarding the safety of some of the vaccines, and then we will definitely end our discussion. So this is a simple question. Maybe we can have a discussion. You must search that which disease do you think was first eradicated from the world by vaccination program, right? So you should try to find out this answer, right? Definitely, this is the only disease which is eradicated from the world, right? In the across the globe, right? So now let us just have the last point of discussion of today's webinar: adverse event following immunization, right? Because as I told you, these vaccines are intended to be taken by normal persons. So such a case, safety is prime importance, right? All of us, we are having that mentality, yes, that we should go for vaccination, but then down the line we have some type of that doubt that whether after taking the vaccines, do we have is there a chance of adverse effect? Yes, definitely there is a chance of adverse effect, right? That's a common thing, right? We'll categorize some of these adverse events, and then we will try to make the discussion on the COVID-19 vaccines also, right? So any untoward medical occurrence which follows immunization, but it's possible that it does not have a necessary causal relationship with the use of vaccine. What is causal relationship? Let's say, for example, if I give ABC drug or vaccine, and then is very clearly associated, it is not a probable, it is not a possible, it is a clear-cut relationship that arisal of adverse event with the administration of that vaccine, then you call it causal relationship. So it is not possible that if a untoward medical occurrence has been produced, it is not necessary that a causal relationship is possible. Then also you categorize it as an adverse event following immunization. So there are different categories of AEFI, that vaccine product related reaction that is related to the vaccine formulation. right? There is a problem with the vaccine formulation. So it's an inherent type of an adverse event which will be observed in all of it or most of the population. Say for example, some of the COVID-19 vaccines, they were reported to produce thromboembolism, right? Clots, right? They were thought to increase the such type of population. Definitely now it has been ruled out. It was reported with the Covishield, and now it is 1 in 10 lakh or 1 lakh. It has been reported like that by European Union, right? By EU, and definitely it has been categorized as a safe, right? You can say as a risk to benefit ratio. But such type of adverse effect related to the product definitely is the vaccine product related reaction. Second is vaccine quality defect related to the reaction, right? So in such a case, adjuvant problem or maybe manufacturing defect you can consider. Immunization error, these are preventable errors, right? Third, that is immunization error. Say for example, while administering the vaccine, while storage of vaccine, such type of an error can produce, and that definitely these are preventable type of an error, and this may be worked on, right? Sometimes anxiety related to the immunization may produce the vaccine, may produce the adverse effect. This is again a common, right? And then sometimes coincidental level, if any adverse effect which is not, you can say, fitting in any of these 4 categories, right? That is first 4, then you call it as a coincidental type of an events, right? Again, these all categories you can classify in a mild, severe, or that morbidity related type of an adverse effect, right? So some cases the effects are, say for example, pain or fever related to the vaccination, so those are mild, right? Then sometimes it may lead to mortality, then it's a serious type of an adverse effect, right? And then if it is not related to morbidity or mortality, but still it is the adverse effect are higher as compared to fever and that pain at the site of it, then you call it as a severe type of adverse. So 3 categories are there: mild, then you have severe, and then most one is a serious, right? Serious adverse. So just like drug, same type of protocols are there, same programs, regulatory bodies are there. That is your pharmacovigilance program under the aegis of CDSCO. It is working to monitor the adverse event. You have passive type of a process where the practitioner gives the advice to the district immunization officer, and then through the channel of pharmacovigilance program you can report the adverse effect related to the immunization, and you can definitely go for a documentation and reporting of such type of thing. This definitely takes a lot, it will take lots of time, so I'm not going in detail, but as I told you, it is the pharmacovigilance programs that you need to consider for such type of documenting and reporting of adverse event, right? So I think I have tried to cover all the details which I mentioned in the learning objectives, right? Again, I am thankful to the organizing committee. I am thankful to Dr. Anand, Dr. Lalitla Madam, Dr. Abhay Dharamsi Sir, and all that trustees of Parul University to provide me the platform for sharing my knowledge. And thank you all participants, right, for your kind listening for this topic. Thank you. Over to you, Anand sir. I definitely welcome if there are any questions related to the topic.
Hello. Yes, participants are requested to ask question if any doubt related to the webinar.
Good afternoon, sir.
Yes,
can you hear me?
Yes, yes, definitely.
Sir, I'm Dr. Vivek,
yes,
working as associate professor.
Yes.
Sir,
yes.
I have got one question. That is, what is the difference between seroconversion and seroprotection?
Sir, it is possible that even after the generation of antibodies, if antibodies are short-lived, or sometimes antibodies that do not have the avidity for antigens, then there is a chance that even after seroconversion, you don't have effective protection. Sometimes you have the pathogens which is continuously variable. So those type of variable type of antigenically variable type of pathogens, there also once antibodies are generated, still there is a chance that a newer strain of a disease is available and it may lead to a disease. So there, seroconversion means you have that antigen, say for example in your body, but then it actually gets converted in disease. Let's say for example in case of HIV and AIDS, we just have a process of seroconversion. HIV infection is there in the person, but then actually complications of reduction of CD4 plus cell count is taking a long time. When it takes time, you call it as a seroconversion. So there is a huge difference. There is a long time, lag time between the because the inoculation of an because there are presence of pathogen inside the body and the observation of the symptoms. Right? That's why we say that AIDS is a different, HIV infection is a different. That's what we normally say. So similar thing you can observe for the vaccine also. Once you've given vaccine, seroconversion will take some time.
Okay. Thank you.
Yes, other participant, any doubt or question you can ask to the sir.
Good afternoon, sir.
Good afternoon.
Sir, if someone is on immunosuppressive therapy, then he or she may be safe to use the vaccine.
Yes, definitely. But in that cases, separate guidelines are given. But yes, if a person is on immunosuppressive therapy, vaccination response definitely will be poor, right? Because without TH cells, cellular immunity as well as antibody mediated immunity will be not sufficient to generate the immune response. Even this is a study being conducted right now by ICMR, where the person who are having autoimmune disease and those who are immunosuppressive therapy, what is the response of the COVID vaccines, right? Right now there is a study going on, and definitely will come out with some results, right? But yes, if you are using immunosuppressive therapy, response is going to be modified. This is especially in case where you have a long-term use of high doses of immunosuppressants, then and then only. Short-term use of steroids, or it is not going to produce much difference.
Okay, okay. Thank you very much, sir.
Okay, I think now there are no more questions. Okay, so link for filling the feedback form and getting an in-certificate is shared in this chat box, as well as we have shared in PU webinar Facebook page, and with the chat box we have shared the link. So thank you, Devang sir. I would like to represent a zest of your nice lecture on the development of vaccines. So it included basics of vaccination, types of immunity, molecular mechanisms of vaccinations. You also provided the introduction to herd immunity, further development of vaccines, their clinical trials, including step-down approach, and also the further challenges and safety. So once again, we are very thankful to you for covering all the aspects in this short term here. So as a PU Parul University organized such webinar for the benefits of students and faculty members through this type of the virtual platform. So as a token of appreciation, we would like to present an e-certificate to you. So just give me the permission to share my screen.
Yes, definitely, sir.
Yes, sir, my screen is visible.
Yes, sir. Yes, sir.
Okay, so this e-certificate
Thank you very much, sir.
from our Parul University to you. We will share this certificate through you to mail.
Thank you, sir, and thank you again for providing the opportunity to share my knowledge. And I will be happy to share knowledge in future as well.
Definitely, sir. Thank you. Thank you again.
Thank you, sir.
So with this, we are concluding the today's webinar session. Thank you all once again.